Location, CNN, central nervous system. Function, produce myelin sheet. Myelin, produce myelin sheet. Has anybody ever heard about myelin sheet or myelin? What is it?
It wraps around the axon and it like, I can't think of the word, it keeps it
Insulated.
insulated. There you go.
True. So, function of the myelin, okay? Myelin basically is a fatty tissue. Well, you can't call it tissue, sorry. Fatty substance. Okay? It basically wraps or encloses the axon. Okay? The axon. So, if I have, basically imagine my arm is being the axon and my long sleeve would be the myelin sheet. Why do you wear sweaters in winters? Why don't you wear short sleeve or sleeveless? Because it's cold outside, right? What does the long sleeve do? Okay, it insulates you, right? Same thing. Now, in physio, you will learn what is the purpose of insulation. I'm not getting into that, okay? But there are certain axons in our body which are myelinated, some are not. Even in myelinated, you have heavy myelinated axons, you have low myelinated axons, or you have unmyelinated axons, okay? And different locations, different places, different degree of myelination. The cell that does that is oligodendrocyte. Now the characteristic of an oligodendrocyte is basically a cell that is on the top, and it basically one oligodendrocyte can myelinate up to 100 axons. So you could see this is one oligodendrocyte, it's myelinated this axon, it's myelinated this axon, so forth. Does that make sense? So it's not enclosed within the myelin sheet. Okay, it sits on top and it sets out its projections. Okay, and that projections will go and myelinate the axon. One oligodendrocyte can myelinate up to 100 axons at a time. So you can see right here, here's oligodendrocyte, it's sending it out projections. The projection goes and wraps the myelin, or wraps the axon. Now Schwann cell. Location CNS. Oh, sorry. Peripheral nervous system. Function is to produce myelin sheet. So oligodendrocyte and Schwann cell basically have the same function, but they're located in two different areas. It's like saying there's a family business. Mike and Carl run a family business. Mike lives in Arizona, Carl lives in California. Okay? They run the family business, which is to produce myelin sheet. One does it in the CNS, one does it in the PNS. Now, one characteristic difference is one Schwann cell can only myelinate certain segment of an axon, not even one complete axon. Okay, and they actually get enclosed within the myelin sheet. So here's the axon. This is a Schwann cell. This is a Schwann cell. Okay, this is a Schwann cell. So this one will secrete the myelin sheet. Okay. Well, I can't do around, but and you see it gets enclosed within the myelin sheet. This one will produce myelin sheet, and it gets enclosed in it. Does that make sense? So right here you could see, here's a Schwann cell, and it starts creating the myelin sheet, and it gets enclosed within the myelin sheet. Okay, so it only creates one segment. So this is saying this segment right here. you have another Schwann shell which does this segment. Okay? Make sense? Yes.
So it like wraps itself around its own product?
Yes. It wraps itself within its own product. Oligodendrocyte sits on top, able to myelinate multiple neurons. Where Schwann shell can't even do one neuron. It only does segment of a neuron. I'm not going to get into what a now, do you see over here there's a gap? There's no myelin sheet right there. This gap, okay, so let me draw it again. So I have an axon. So, red was the axon, green is the myelin sheet, blue is the Schwann shells. And as you can see, it's actually getting close within it. But right here, there are these gaps, right? Which are not myelinated. Okay? Those are bare regions of an axon. Okay? You don't call it bare axon, you call it node of Renwier. Node of Renwier. Okay? So node of Renwier are these black areas that I'm drawing right now. It's a bare axon.
What is the green again?
Green is the myelin sheet. Red is the axon. Green is the myelin sheet. Blue is the Schwann shell. And the black are the bare areas of the axon, where the axon is actually exposed. And you don't call it bare axon, you actually call it node of Renwier. Node of Renwier. Okay? Does that make sense? In physiology, you'll learn the importance of node of Renwier. So it's saying I have a long sleeve, okay, all the way, but I have little holes right here. Where the holes are, that's where I would be able to see my skin, correct? That would be a node of Renwier. Okay? Any questions so far? Now let's talk about a synapse, the different type of synapses. Synapses is basically communication between a neuron and another structure. There are three types based on the interaction. So you have basically a neuron, and there's a synapse, and then you have another neuron, which are the dendrites. Okay? Which you just call it a junction is between a neuron and a neuron, or synaptic. Okay? Then you could have a synapse between a neuron and a muscle cell, which we already learned about, which is called the neuromuscular junction. This is just called synaptic cleft, or synapse. Third one is a neuron, and you could have a junction between neuron and glands in our body. Does this make sense?
Is it called something specific?
Yeah, I don't remember the name. I forgot what it's called, sorry. We don't usually use this one that much, so I forgot exactly what it's called. I don't, I think your book just calls it neuro to a gland, but I think there's a specific name as well. But you could just call it neuro to gland. This is the same photo from your book. So if you find this photo, it will be now, synaptic communication. A nerve impulse, basically how does communication occur? Okay, synaptic cleft is there for communication. What actually happens, an electrical impulse is changed into a chemical one. Then a chemical impulse or chemical signal is changed to once again into electrical signal. Okay? Does that make sense? So let's say let's say every single table is a neuron, okay? Now, basically, I'm sitting on this table, I started off, okay? So basically, let's say I whisper something into her ear, okay? She hears it, she whispers into her ear, and so forth. So this is an electrical signal going from within a neuron, there's electrical signal. Between neurons is a chemical signal. Okay, electrical signal occurs on a neuron. Chemical signal occurs between neurons. Okay? So I say something into her ear, she says something into this person's ear, so forth. Now the signal has to jump from this neuron to the next. How does basically the last student, okay, how does, if he can't whisper, let's say the tables were really far apart, he could not whisper into that person's ear, okay? How do you think he would, he can't say it out loud, how do you think he would pass the message from this table to the next? A note, right? He will write it down, he fold it up, and he will hand it. Because the hands are long enough, both of them extend their hand out, they're able to pick it up. Now that's a chemical one, does that make sense? So on an axon, it's an electrical signal. From axon to another structure could be axon, another neuron could be a muscle, something, it becomes a chemical one, which is the neurotransmitters. Okay? So, a nerve impulse triggers events that transfer information across the synapse. Action potential, the electrical signal is called action potential. Okay? Electrical signal, or the electrical impulse is called action potential. So, action potential occurs on the neuron. As soon as it arrives at the axon terminal, that is converted into a chemical. Remember the neurotransmitter is released, right? That neurotransmitter being released and binding to the postsynaptic membrane, that was a chemical signal. As soon as that neurotransmitter binds to the postsynaptic membrane, that chemical signal now, once again, would be changed into electrical signal because action potential will be generated again on the neuron. Okay? Does that make sense? So as soon as he receives the note, he will read it and whisper into this person's ear. She will get it. Now, again, she will write a note, send it over. Does that make sense?
You said it was called action potential?
Axon,
Or
axon.
Oh, axon.
No. Action potential. That's the electrical signal. Or the electrical impulse is called action potential. Yes. Well, it causes a neurotransmitter to be released. Yes.
Once the action potential reaches an axon terminal, becomes a chemical signal.
Yes, because what happens, action potential actually starts on an axon hillock. As soon as it arrives on the axon terminal, it causes the neurotransmitter to be released. Now the neurotransmitter is released within the synaptic cleft, or the neuromuscular junction. And that chemical signal, now, which is the neurotransmitter, is going to go and bind to the receptor on the postsynaptic membrane. And as soon as that receptor binds, basically now that chemical signal, once again, is changed into an electrical signal, because a new action potential will be generated on that neuron. Does that make sense? Once again, that new neuron generates action potential on the axon, starting from the axon hillock, will go all the way to the axon terminal. Again, it will cause the neurotransmitter to be released. Neurotransmitter will release, bind to receptors on the postsynaptic membrane. Once again, that signal now is again turned into electric. Yeah.
So the electrical impulse is just called an action potential, and then the chemical signal is called a neurotransmitter?
No. I mean, that electrical impulse is basically action potential. Okay? I have not told you what these are yet. I'm just trying to explain something else. Yeah. So basically, this is something else which I haven't gotten to explain yet. I'm just trying to tell you whenever I say there's an electrical impulse or a nerve impulse, it's actually saying action potential. That's what it is. Okay. Transfer process accomplished by two ways. One, synapse, electrical, this is transmission. Synaptic transmission involves a neurotransmitter, which is a chemical signal. Let me see for a second. Okay, sorry. So this is this process completely, I need to read myself. Other part becomes too basic and you forget their names. Okay, so synaptic transmission is just a process where communication is occurring between one neuron and something else. Anytime it involves a neurotransmitter, it's called a vesicular transmission. And if it involves flow of ions, it's called non-vesicular transmission. Does that make sense? Because remember, in the axon terminal, the neurotransmitter was found within a vesicle, right? Therefore, if it involves a neurotransmitter, it's called vesicular transmission. And other one is non-vesicular transmission, okay? Or vesicular synapse or non-vesicular synapse. Okay? Yes?
So is synaptic transmission the same thing as the
Synaptic transmission is basically, there's two types. Synaptic transmission just tells you that there is communication going on between a neuron and something else. Now, how is that message, how is the communication occurring? It can be occurring due to neurotransmitter being released, or because there is actually ions being released, okay? If that communication is occurring because the presynaptic membrane releases a neurotransmitter, it's called the vesicular transmission or vesicular synapse. Okay, vesicular transmission or synapse. Some books will say vesicular transmission, some books will say vesicular synapse. If the presynaptic membrane does not release a neurotransmitter, but it actually releases ions, okay, that's a different one which we don't usually even talk about that one in physiology because that's more PhD. Mostly in the physiology we'll talk about the vesicular transmission, okay? If it involves the flow of electrons, not electrons, flow of ions between two membranes, then it's called non-vesicular. Does that make sense? Okay? Both of these are synaptic transmission because the two pre and post are still communicating with each other. Does that make sense? Okay. Yes? Okay. Now we're going to talk about action potential. As I said, that was the electrical impulse which was generated, right? About a communication for the nervous system. Because this is the electrical impulse that occurs on the axon. So what is it? Now, before I explain what is it, you have to understand that axon is part of a cell, right? It's a neuron. Inside the neuron, we have electrolytes, or we have ions. Outside the neuron, we also have ions. And basically, ions are just moving in and out freely, okay, at certain time. Now, certain ions are inhibited to enter the cell, certain ions are inhibited to leave the cell, okay, leave the neuron or enter the neuron. Now, certain things will trigger where there's a rush of electrons inside, not electrons, rush of ions inside, rush of ions outside, okay? Does that make sense? So basically, what we do, we measure the charge or the electricity or the potential of the nerve or that axon. Does that make sense? Okay, right now, if I place electrode and I check, what is the conductivity of this class? There's nobody going in and out, right? So it's low, correct? But what if all of a sudden students start leaving, students start entering? Doesn't that change the conductivity? Okay? So what is the action potential? You're just measuring the conductivity. Okay? And you're measuring it and you're plotting basically like a graph. So action potential is a wave-like appearance. Because why is it wave-like? Because it tells you all of a sudden certain ions are entering, so the charge of the membrane becomes too positive or becomes negative. Does that make sense? Okay? Action potential is just a wave-like appearance because there is rapid influx or rapid efflux of ions, and you're just measuring the conductivity. Okay? I know I'm not good at it. You'll understand it a little bit afterwards. But do you guys understand what I'm trying to say? Okay? Influx and efflux.
So it's just a measurement of what is going, or how much is going in and how much is coming out. Okay.
Exactly. And we are measuring that and we plot it, and it looks like a wave-like appearance. Does that make sense? Does that make sense? Okay, what is action potential? It's a wave-like appearance, okay? So the appearance is a wave-like appearance, okay? Doesn't it look like a wave? This is one action potential, and it looks like a wave, right? Because it's just measuring the conductivity of ions entering or exiting, okay, an axon. Not the cell body, not the dendrites. We're only measuring the axon. So where is this occurring? Where is the location? Axon, okay? So location is axon. Because we're only measuring the conductivity of the axon. Does that make sense to you guys? Okay. So that is electrical impulse. Remember I told you electrical impulse starts at the axon hillock and goes towards the axon terminal, correct? That's electrical impulse. And that electrical impulse occurs because there is movement of ions going in and out. It's like how a battery works, right? Battery, constantly ions are moving, are flowing, right? And it causes you to work, okay? Okay. Action potential. What triggers its occurrence? Right now, just know gradient potential. Now in physiology, this goes extremely in detail. Okay, right now I'm just setting up certain basics, so when you go into physiology, you won't be completely lost. Okay. What triggers its occurrence? Gradient potential. I'll tell you what that is a little bit later. Now, action potential has three phases, and they occur in this particular order. Depolarization, followed by repolarization, followed by hyperpolar. Action potential have three phases. Depolarization, followed by repolarization, followed by hyperpolarization. They occur in this order, and that's what you have to know. What is the result? Why do you have action potential? So what was the result of action potential or nerve's impulse arriving at the axon terminal? What was the purpose? The purpose of the electrical impulse arriving at the axon terminal was so the neurotransmitter can be released. Okay? Release of neurotransmitter. Okay? So that is the purpose. This is why the axon conveys information. Remember dendrites picked up the signal? What did the dendrites pick up?